This is the genetics review that we did in class today in AP Bio, and I'm going to walk you through all the problems. So we're going to start by walking through some basic terms and different kinds of crosses that we covered in this chapter. Sometimes these crosses get confused with one another, so I want to make sure that you are clear on the differences between each one. So dominance is the basic cross that we learned from Mendel. This is where, for example, let's say that big B represents an allele for black, and let's say that little b is going to represent an allele for white. Big B, and big B, little b, if this is a trait that shows dominance, would both show the trait of black, and then little b would be white. In codominance, what we see is that neither one can cover up the other. So, big B would be black, and little b would still be white, but this time big B would result in spots or speckles of both black and white. In incomplete dominance, we see something somewhat similar. We see that our big B would be black, and our little b would be white. But in incomplete dominance, rather than seeing spots, we would see an intermediate phenotype, so we would see something like gray. Pliat Ropey is a special situation that involves still just a single pair of genes. So we have only one pair of genes. But what happens is these genes can affect more than one phenotypic characteristic of the offspring. For example, in class we talked about dwarfism. If somebody is big D, little d, and they have the gene for dwarfism, it doesn't just make them short. Along with that comes a whole series of characteristics that are part of dwarfism. Another example of this, which was in your Mastering Biology assignment, was with tigers. They said that in tigers there was a gene that showed Pliat Ropey, and that when you crossed them, that big B coded for the tiger to be orange and regular eyes, and that the little b gene coded for them to be white and also coded for them to have crossed eyes. And then there was a question that was a follow-up to this that said, so if we crossed two heterozygous ones, what would be the chances of them being white? Well, this is a regular cross, big B with big B little b, and we're treating this just like a dominant recessive, so little b would code for white, and therefore 25% or 1/4 would be white. But then there was a follow-up question, and it said, of the ones that were white, what percentage of them would have crossed eyes? And this was somewhat of a trick question, because if white and crossed eyes were both caused by the same gene, then that would mean that 100% of the ones that were white would have crossed eyes, because the gene causes both of those characteristics. So that is Pliat Ropey. The last two, polygenic inheritance and epistasis, are very similar to one another. So let's go through the difference between them. In polygenic inheritance, what we see is an additive effect, meaning the more capital letters you have, the darker you would be, the taller you would be, etc. So for example, in one case, we could say that, let's say that big A, big B, big C here, coded for you being black. All capital letters. Now, if this is polygenic inheritance, what would happen is, if you had 5 capital letters instead of 6, then maybe you would be deep gray. And if you had 4 capital letters, maybe you'd be a medium gray, etc., all the way down to having no capital letters, and that would make you white. The important thing to note here is that it could be any 5 capital letters. So it could be this, big A, little a, big B, big C, big C, or big A, big B, big C, big C. In other words, in polygenic inheritance, these two would look alike because we're just counting how many copies of the dominant form we have, and it just adds together. Now with epistasis, we see something different. And in fact, the next slide is going to, actually the next couple slides are going to be examples of epistasis. But basically what happens with epistasis is that you have 2 or more genes and they combine to produce a trait. Let's go to the next slide and you'll see what I mean. Alright, so this is actually a really good example of epistasis. In parakeets, we have 2 genes. There's a Y gene and a B gene. And the specific combination of Ys and Bs gives us 4 colors that we can have. We can have, if there's at least 1 capital Y and 1 capital B, we have green. If it's at least 1 capital Y, but little b, they're yellow. Lowercase y's and at least 1 big B, they're blue. And that shouldn't be there. And then lowercase y's and lowercase b's, and they come out white. If this was polygenic inheritance, it wouldn't look like this. What you would see is, for example, big Y, big B, and big Y, big B, little b, in polygenic inheritance would look exactly the same, because they both have 3 capital letters. That's polygenic. But this is not that. Notice that in this case, it's the specific combination. It's not just 3 capital letters. It depends which ones they are. Alright, so let's look at a cross involving epistasis. So they want you to cross 2 heterozygous parakeets. So we are going to assume that we are crossing big Y, big B with big Y, big B. Now, you could solve this with a Punnett square, but the easiest way to solve this would be to separate out our Y's and our B's, and then use the product rule to figure out our chances of the 4 different genotypes here, green to yellow to blue to white. So, if we cross just our Y's, which would look like this, we're going to get a Punnett square that looks like this. And when we cross our B's, in this particular case, we're going to get pretty much the same exact Punnett square. Now, the interpretation is what matters here. Let's take a look at what we need in order to have a green parakeet. So, in order for a parakeet to be green, we need at least 1 big Y and at least 1 big B. So, what are the chances of at least 1 big Y? This one, and this one. So the chances of at least 1 big Y are 3/4. And then the chances of at least 1 big B are also 3/4. We multiply these together, that's the product rule, and we get a 9/16 chance of green. Now we're going to repeat the same process for yellow. So in order for a parakeet to be yellow, we need at least 1 big Y, and then we must have little b. So the chances of at least 1 big Y, again, are 3 out of 4. And the chances of little b this time are going to be 1 out of 4. So our chances of yellow are 3/16. We can repeat this now for the other 2 genotypes. In order to be blue, they have to be little y, and then at least 1 big B. That's going to be a 1/4 chance of little y times a 3/4 chance of having at least 1 big B, and that's going to give us a 3/16 chance of blue. And finally, for a parakeet to be white, it has to have little y, which there's a 1/4 chance of, and little b, which there's also a 1/4 chance of. So 1/4 times 1/4 gives us 1/16. And we should be able to double check ourselves and make sure that this adds up to 16 out of 16. So we have a 1/16 chance of white, plus 3/16 chance of yellow, plus a 3/16 chance of blue, and a 9/16 chance of green, and that adds up to 16 out of 16. Now they want you to write here the ratio of green to yellow to blue to white. We need to make sure our denominators are the same, which they are. So our chances of green were 9 out of 16. Chances of yellow were 3 out of 16. Blue was 3 out of 16. And white was 1 out of 16. So writing this as a ratio, our ratio would be a 9 to 3 to 3 to 1 ratio. And you may remember that we talked about this, that in a basic dominant recessive cross, when you cross two heterozygotes, you often get a 9 to 3 to 3 to 1 ratio. All right, building on our last problem, this problem is related. So they say that you cross a blue parakeet with a white one, and they want to know what are all of the possible colors that you can get. Now, since they don't tell you the genotype of the blue parakeet, what you should do is assume whatever genotype would give you the most leeway. In other words, a blue parakeet could be little y, big B, or little y, big B, little b. Since we're trying to see the most possibilities, this parent can give us more possibilities in offspring than our homozygous parent. So we're going to, for the purposes of finding what's possible, we're going to use our heterozygous one. So when we cross our little y, big B, little b, with a white one, which would be little y, little b, let's look at our possibilities one at a time. Could this give us a green parakeet? Well, if we go up to green, you're going to see up here at the top that we need for green at least one big Y. And we don't have that. We have little y for both parents. So green is not possible. And when we look at yellow, we have the same problem. We only have little y's, so that's not possible. For blue, we would need a little y, which is possible, and then a big B with anything. And that is definitely something we can get. And finally, for white, we need little y, little b. And again, we can get that. Each parent has a little y, and each parent has a little b. So these would be the two possibilities from this cross, blue parakeets and white parakeets. I added this question to illustrate how you would figure out the number of possibilities of what a parent can donate after meiosis. So, the question is, what would be the dimensions of a Punnett square if you were going to cross this parent with this parent? Here's our rule we learned in the last chapter, that the number of possibilities was 2 to the n, where n was the haploid number. Now we need to expand on that a little bit. In other words, if we had little a, little b, there would be 2 pairs of chromosomes, so it would be 2 squared. But are there really 2 squared or 4 different possibilities here? No, in fact, there's only 1. They can only give little a, and they can only give little b. So when we use this formula for this chapter, what we're going to revise this to be is 2 to the n, where n is going to be the haploid number or the number of pairs, but we're only going to include pairs that are heterozygous. Because if they're homozygous, there is no choice involved. They can only give that thing. So to figure out the dimensions of our Punnett square, let's look at our first parent. This parent has 4 heterozygous pairs of chromosomes. 2 to the 4th gives us 16. In other words, for our Punnett square, we would need 16 boxes across, or down, it doesn't really matter. For our other parent, notice that we only have 2 heterozygous pairs. So now we've got 2 squared, which is 4. So the other dimension of our Punnett square would be 4. And 16 times 4 gives us 64, so this would actually be a 64-box Punnett square in order to represent all the possible genotypes of this cross. Alright, this is a harder example involving epistasis. So in this case, we're talking about corn. If you have at least one big P, the kernels will be purple. And if it's little p, the kernels are red. However, there's a completely separate gene that basically overrides that first one. If they carry a capital I, then they have a color inhibitor gene forcing the corn to be white. And if they're little I, then they don't have that gene, or they have the recessive form, and then color will be allowed. So what they want to know is the ratio of red to purple to white if we cross 2 heterozygotes. So we're crossing big P, big I, with another one that's big P, big I. For the record, both of these parents would actually be white because they both have the color inhibitor gene that's going to prevent their purple color from being expressed. Alright, we're going to separate out our P's and our I's, just like we did for the other problems. And then we need to look at the possibilities that will give us each of those 3 phenotypes. So let's start with red. So in order for corn to be red, we know it has to be little p, which is basically a 1/4 chance, and it can't have the color inhibitor, so it has to be little I, which there would also be a 1/4 chance of. Therefore, the chances of red corn would be 1/16. Now let's look at purple. In order for corn to be purple, it would need to have at least one big P. So basically, there's a 3/4 chance of that happening. We also would have to have little I again, our color inhibitor. When we multiply that one out, we get 3/16. Now technically, since there's only one other option, we could actually just add this up and say, okay, that makes 4/16, therefore the rest of them would have to be white, in which case that would be 12/16. But let's just say that you didn't know how to do that. Let's see how to calculate the chances of white. And this one is harder. So here's what's going to happen with white. We know for the I gene, in order to be white, big I anything is going to be white, and that's a 3/4 chance. And what about on the P gene? Well, the truth of the matter is it doesn't matter what they're carrying on the P gene. It's irrelevant, because whether they're big P, big P, or little p, if they carry big I, they're automatically going to be white. So how do you represent that the possibility doesn't matter? You can just represent it with a 1, meaning 100% of the time they're still going to be white. When you multiply this out, you get 3/4. 3/4 times 1. But be very careful when they want the ratio. You can't put that it's a 1:3:3 ratio. That would be wrong. Why? Because that ratio has to do with out of 16 individuals, 1 is red. And out of 16 individuals, 3 are purple. So what we need to do is figure out of our 16 individuals, how many would be white? Well, 3/4, if we want to change that into 16ths, 4 goes into 16 4 times, and 4 times 3 is 12. Therefore, our ratio would be a 1 to 3 to 12 ratio. All right, our next problem is a blood type problem. Now, blood type shows actually a couple of things. One thing that it shows is codominance, because I, which stands for immunoglobulin, it's the protein on the blood cells, IA and IB are both dominant. They are codominant alleles, forms of the gene. But this is also an example of something called multiple alleles, because there's not just two alleles, there's a third one, little I, and little I is recessive. So the combination of these alleles will give us our blood type. For example, IA, little I, would be blood type A, because little I is recessive. IA would also be A. IAIB would be AB. And the only way you could be O would be you'd have to be little I, little I. So in this question, we're presuming that 3 couples each had a baby in the hospital on the same day. Couple A, we know their blood types were both A. Couple B, one was A and one was B. And in couple C, one was B and one was O. The nurses mixed up the 3 babies. So we're going to try to match the babies to their correct parents. Now keep in mind, some of these, for example, looking at couple C. Couple C could actually be the parent to more than one of these babies. Couple C would have to be IB little I. We're going to assume heterozygous again, because that gives us the greatest range of possibilities. And then they're also, the other parent is O. So this is what our cross would look like. So what kind of kids could they have? Well, they could have a kid that's type B, or they could have a child that's type O. Well, there's 2 babies here. There is a B and there is an O. However, if we're assuming those were the only 3 babies born that day, we should be able to use process of elimination to figure out which of those 2 babies belongs to them. So let's look at couple A. So in couple A, we have 2 parents that are A, IA little I, IA little I. Again, you may say, why did I assume they were heterozygous? Because that gives us the greatest range of possibilities. And by the way, if we don't assume they're heterozygous and they're both IA, IA, then the only kind of kids they could have would be type A kids. And if you notice our results, there aren't any type A babies. So they have to be heterozygous. So if we do this cross, we see that they can have type A children. This would also be a type A child. This would be a type A child. And then they have a 1 in 4 chance of having a type O child. Now, since none of the babies were type A, that would mean that number 2 has to be their child. It's the only other option. Now that you know that couple A goes with number 2, that would mean that couple C, that I showed you first, has to go with number 1. We know this because couple C, nobody has type A. And the baby number 3 is AB. Now we can verify that we're right by doing our cross with couple B. So let's say that couple B is IA little I and IB little I. If we do this cross, we can have a child that's AB, a child that's B, a child that's A, and a child that's O. In theory, they could be the parents to all 3 of those kids. But since we already know the parents of baby 1 and baby 2, these have to be the parents of baby 3. And they're the only parents that could give birth to an AB child, so they have to be the parents of baby 3. Okay, for our last slide, we're going to go back and look at how to do a chi-square test using a genetics cross. So in the past, we've done chi-square tests, but we always did them with a null hypothesis, meaning we made a prediction that there would be absolutely no difference in the number of mice in each chamber or whatever. So we would just take the total number of observed offspring, and we would simply just split it into even groups. But you don't do that with a genetics chi-square test. With a genetics chi-square test, they're telling us that they think our original cross was big B little b and big B little b. They also tell you that there are 3 phenotypes in this cross: black, gray, and white. Hopefully you recognize that this would be an example of incomplete dominance, because you're getting an in-between phenotype in the heterozygote. Alright, so you will get a table that looks like this, and they're providing you with the observations that were made in the lab. You can't make those up, you have to be told that. So, out of 200 babies that were born, there were 55 black, 110 gray, and 35 white. Now what you need to do is figure out how many we would have expected to be black, gray, and white. So if we're correct about our Punnett square, we would be crossing big B little b with big B little b. And that gives us a 1/4 chance of black, a 1/2 chance of gray, and a 1/4 chance of white. So now what we need to do is figure out how many that's actually going to be in our table. So if we have a 1/4 chance of black times there were 200 offspring, that means we would have expected of our 200 offspring, 50 of them to be white, or black, sorry. For gray, it was a 1/2 chance, again, times 200 offspring total, and we would have expected 100 of them to be gray. And for white, it's the same as it was for black. It's a 1/4 chance of white times 200 offspring, and that gives us 50 that we would expect to be white. All right. Our next step is to now calculate chi-squared. So chi-squared, I'm going to cover this up because you don't really need it right now. Chi-squared, you'll be given the formula for it. It's observed minus expected squared over expected. And you'll see this sigma in front of it. That means it's the sum of the observed and minus expected squared over expected for all 3 of our traits. So we need to fill in a chi-square for this, a chi-square for this one, and a chi-square for this one, and then we will add those together to get our final chi-square, which we will then work with. Alright, so again, you can use a calculator and you're provided with the formula, so I don't want to spend too much time, but the first one would be basically 55 minus 50 squared over 50, and you end up getting 55 minus 50 is 5. 5 squared is 25, and 25 over 50 is 0.5. Our second one, 110 minus 10 is 10. 10 squared is 100. Divided by 100, we get 1. And for our last one, 35 minus 50 gives us 15. If we square 15 and then divide that by 50, we get 4.5. So if we add these up, we get a total of 6. And this is the number that we need to work with. For our last step, we are going to go to this chi-square table down at the bottom. And this is the one that's provided for you on the AP exam. So across the top here we have degrees of freedom. This is going to tell us which column we're supposed to be looking at. The degrees of freedom is the number of phenotypes minus 1. In this case, we had 3 phenotypes, and they were right here, black, gray, and white. 3 minus 1 makes 2, so that means we're going to be looking at column 2, right here, 2 degrees of freedom. All right, I don't know why they include this, because we're always going to be looking at the 5% cutoff, which is what the 0.05 refers to. So this is our cutoff number, 5.99. 5.99, that means that if you get a number larger than 5.99, there would be a less than 5% chance, that's what the 0.05 over here on the left means, a less than 5% chance that that should happen. Meaning your data is so far off your predictions that something might be wrong. You can't support your prediction. So ours fails. We got 6.0, which is a bigger number than 5.99. What would be the reason this could fail? Well, maybe you had a contaminated sample. Maybe you have someone who can't count. Or maybe, since this is a genetics cross, maybe this is not the correct pattern. Maybe this trait, we didn't realize it, but maybe this trait is controlled by multiple genes. Maybe there's another gene. Maybe there was a mutation. But all we know is we have to reject this pattern. This does not fit with what we predicted. And therefore, we have to say that we would have to repeat the experiment or come up with another hypothesis of how these results came about.